So good morning, grade 12 students. In this session, we're going to continue with our chapter about the nervous system, chapter 12 about the neurotransmitters and the medical applications. And in this document 4, we're going to talk about the action of drugs on the synapses. Already in the last session, we talked about the amplification of the nervous system. It's done by 4 steps or 4 different phenomena. We have to provoke or to facilitate or to help or to allow the exocytosis of the neurotransmitters in the synaptic vesicles from the presynaptic terminals. Second, to amplify the nervous system, we have to inhibit the recapture of the neurotransmitter. So there is no recapture, so the neurotransmitter will stay in the synaptic cleft and will be able to amplify the nervous system. Third is to inhibit the action of the enzymes that degrade the neurotransmitter. These enzymes, they are inhibited, so no more function of these enzymes. So we have to inhibit their function so that the neurotransmitter will be able to work and work more and more to lead to the amplification. And also step number 4, or the phenomenon number 4, is to facilitate or to provoke or to help or to allow the binding of the neurotransmitter to the receptors of the postsynaptic terminal. All of these, as I said yesterday, they are used in the analysis, in the interpretation, and also in the conclusions or hypothesis. So all of them, they have the role here. And if you know this, you have an idea about what the conclusion will be, what the hypothesis will be, and the interpret or the indication will be. While regarding the inhibition of the nervous system. Inhibition of the nervous system, first you have to inhibit the exocytosis of the neurotransmitter from their secretory vesicle in the presynaptic terminal. So whenever you inhibit their exocytosis, so there is no function of these neurotransmitters, you have to provoke the recapture of the neurotransmitter in the presynaptic terminal. So you have to inhibit their recapture. Whenever you you have to provoke their recapture. So recapture, so if these neurotransmitters are recaptured into the presynaptic terminal, also you have to facilitate or to provoke the action of the enzymes. So whenever the neurotransmitter binds to the receptor, does its function, so then this neurotransmitter is going to be degraded by these enzymes. So these enzymes, you have to facilitate their function or action in order to inhibit the nervous system. Also, you have to inhibit the binding of the neurotransmitter to their receptors. Whenever you inhibit their binding to their receptors, so their inhibition will be for the nervous system. So these two, amplification and inhibition, they are very important to know them. And also we're going to talk about them in the last question in the probing documents. Yesterday we started about the action of the drugs on the synapses. We said that we have two different types of substances. We have agonistic and antagonistic. Agonistic, they have the same function, or at least one of them facilitates or it makes the function easier for the second. They are called agonistic. If they don't have the same function, at least they don't inhibit their function, the function of each other. This is called the agonistic. However, the antagonistic, they have opposite function. What the meaning of they have an opposite function? For one of them, for example, it leads to the contraction, the other one leads to the relaxation, like the case of acetylcholine and the curare. These two substances, they are antagonistic. One of them, it causes pain, one of them, it relieves the feeling of pain. So these are antagonistic. So antagonistic drugs, they have opposite function, and this is what is known by antagonistic. So here about the antagonist, here about the cocaine. Cocaine, it's extracted from the cola leaves. This cocaine has an important function. They block the reabsorption or block the recapture of the dopamine. So dopamine will stay in the synaptic cleft, which prolongs its effect, feeling of pleasure, of happiness, and so on. So this is the function of the cocaine. It inhibits the recapture or blocks the recapture, and we talked about this before. Also, we move to talk about the amphetamine, and we talk about the benzodiazepines. They are anti-anxiety drugs such as the Valium and others. What do? They activate the fixation of the GABA. It is the GABA. GABA was said mostly, it's an inhibitory. It's GABA gamma aminobutyric acid binding to the receptor as shown in document D. So here we have Cl- channels, receptors with 2 receptor sites. One for benzodiazepines and the other one is for the GABA. So both of them, they bind to this and they are as the inhibitory one, while the amphetamines such as excessive drugs or medicine, they stop appetite, release of dopamine from the storage vesicles, even in the absence of the nerve message. Okay, then we move to talk about dependence or addiction and the tolerance as well. So here the sensation of pleasure when a drug is taken is followed by a desire. So whenever you use the drug, those who use the cocaine, for example, heroin and others, so whenever they took this, they have a feeling or a desire to continue its use. يعني بس يستعملون first time عنده صار فضول ده اللي يستعملون used to this lead to the state of dependence or addiction to this. يعني بيصير مدمن عليهم. However, repetitive use of the drug lead to the states of tolerance, which means that the body was adapted to the doses of this drug and the loss of its effect. For example, إذا نستعمل دواء نحن في ناس كثير بتاخذ بانادول بتاخذ حبة أو حبتين، هدول الحبتين ما بيعملوا معهم effect، يصيروا خلص يتعودوا ياخذوا دواء كثير بانادول حتى يصير ياخذ معهم effect. This is called the state of tolerance. This results by continuous increase of the dose. يصير يزيد الدوز، اخذ حبه ما مشي الحال بياخذ حبتين، حبتين ما مشي حالهم بياخذ ثلاثه، ما مشي حالهم ثلاثه بياخذ اربعه and so on in order to have the effect حتى يخف وجع الراس ونحن كثير شاطرين فيه اللبنانيه بانادول. Okay? Addiction always is translated as a biological adaptation for the product. So addiction is adaptation to a product. The user cannot interrupt the consumption. ما في يوقف. Why? Because هاي incomfortable state. يعني بيكون بحالة لازم يضل ياخذ الدواء. This will lead to the addiction for this. يعني إذا ما أخذ بنادول كل يوم في ناس خلص بتصير مدمنة على البنادول، إذا ما أخذ كل يوم حبة بنادول راسه بصير يوجع، هو بيكون راسه مش عم بيوجع بس خلص نفسية صارت القصة in addition to addiction to this drug or to this medicine. Medical application. The knowledge of the biochemical aspects of the functions of the nervous system has contributed to the understanding of the human behavior. Whenever you have pain, whenever you have emotion, whenever you have insomnia, for example, induced by the medical application of the chemical substances. Example here in document F showing certain medical applications. So this is certain medical application. We're going to use them like, for example, antidepressant molecules. For example, the Curer product that is used by doctors in order to obtain relaxation for the muscles. We said that Curer is used to relaxation of the muscle, inhibition of the muscles. While antidepressant يعني الأدوية ضد الاكتئاب they inhibit the monopeptidic oxidase that degrade the neuromediators interfering in elevating the mood. Antidepressant like the tricyclic substances. Sure, you don't have to study them. But here there are some examples and you are going to summarize them in part 3. So here, part 3 and 4. So here they said compare in a table form the following substances. Curer, cocaine, amphetamine, and the benzodiazepine concerning their site of action and the level of the synapse and their mode of action. This part is very important and you have to know them very well. Curer, cocaine, amphetamine, and benzodiazepine, and they're concerning their action, site, and their mode of action. And also in part 4, we're going to classify the effects in document F. This document F, I'm going to summarize it in few lines in part 4 concerning the role, either the amplifying role of the neurotransmitter or the inhibitory role of the neurotransmitter. This is similar to the notes that we presented in the beginning of the document. These one, they are very similar to this one, talking about the inhibition and the amplification. These notes, they are very similar to part 4. So already we saw part 1 and part 2, A and B, and this table is showing the effects of 4 of the drugs concerning their effect on the synapse and their mode of action. Let's start first with the Curer. Curer, it works at the level of the fixation of the neurotransmitter on the postsynaptic membrane receptor. So here, the fixation on the postsynaptic membrane receptors. What happened to this? As we said before, we said that Curer and acetylcholine ACH, they have highly similar spatial configuration. They are very similar to each other in the structure. They have the similar structure molecule. So they compete and they fight each other in order to bind to the receptor. Whenever they bind to the receptors, the Herra will inhibit the function or the action of the acetylcholine. Thus, it will inhibit even partially or totally, according to the dose and according to the receptors. Either it inhibits totally or partially the action of the acetylcholine. How? Curar, it has a relaxation mode, even though it's an inhibitory role. It inhibits the transmission of the message, leads to the relaxation, and as we said, the Indians, they use their curar at the top of their arrows in order to paralyze the prey and to catch their prey. So QRR is used as the inhibitory neurotransmitter. So it inhibits the function of the acetylcholine, which is an excitatory neurotransmitter. So binding of QRR to the receptor of acetylcholine leads to the state of relaxation, and it blocks the binding of acetylcholine. For example, we have a receptor here. If we have a receptor on the postsynaptic cell, the binding of the QRR or to this receptor will block the binding of acetylcholine. So هيجي الacetylcholine ما حيلاقي مطرح يقعد عليها. So this will lead to the inhibition of the message and blocking the binding sites or the blocking the binding receptors for acetylcholine. So what you have to know that acetylcholine and QRR, they are antagonists. They have opposite function and they compete where? At the level of the binding sites on the receptors on the postsynaptic membrane. Moving now to talk about the cocaine, sorry. Cocaine, it is the second drug. So first we finish about the curar. We said that curar and acetylcholine, they are antagonists. Cocaine, already we talk about the cocaine. Cocaine, it works on the dopamine. What does it do? It works at the level of the recapture process. It works at the level of the neurotransmitter recapture by the presynaptic terminal. As we said before, dopamine is going to be released for a few periods of time. This will lead to feeling of pleasure, of happiness, of ecstasy, and so on. So this dopamine is released for a few period of time, few seconds. Then it's going to be recaptured. So whenever you took cocaine, this cocaine has a role that blocks the recapture of the dopamine. So it blocks the dopamine recapture. So blocking the dopamine recapture, what does it do? Blocking the dopamine recapture will lead to the prolonged effect of the dopamine. So higher feeling of pleasure, of happiness, and so on. That's why those who took cocaine, that use the cocaine, all of the time they're happy, they have a high feeling of pleasure and happiness, and so on. So this is the mode of action of the cocaine. So cocaine, it works at the level of the recapture process and it inhibits the recapture of the dopamine. So this is the second neurotransmitter till now. We talk about the curar and the acetylcholine. So from now on, whenever you say curar, always you have to know that acetylcholine is the antagonist. Cocaine, dopamine, it helps the function of the cocaine. So dopamine, sorry. So cocaine, it helps or it allows or it facilitates or provokes the function of dopamine by inhibiting its recapture. So always whenever you say cocaine, you have to link it immediately to the dopamine. While amphetamine, they work at the level of the release of the neurotransmitter by the process of exocytosis. So amphetamine, what do? They facilitate the release of dopamine. So they help, they provoke, they allow the release of dopamine from the presynaptic neuron. So they have a role in the facilitating the release of the dopamine from the presynaptic terminal. Moving now to the benzodiazepines that are working at the level of the fixation of the neurotransmitter on the postsynaptic. So they allow the fixation of the GABA. GABA, gamma aminobutyric acid, that has an inhibitory role. So these four types of drugs, they are very important and you have to know them. So whenever you talk about curar, immediately you have to link it to acetylcholine. Whenever you talk about cocaine, immediately you have to link it to the dopamine. Whenever you talk about amphetamines, you have to link it immediately also to dopamine, and also benzodiazepines, you have to link it immediately to the GABA. This table is very important, and you have to know this drugs, their effect on the synaptic function, and also their mode of action. And this will help you a lot in the exercises and in the applications. Part 4, talking about the disorders that occur whenever we have amplifying effects, increasing the effect of the neurotransmitter. What are they? The disorders, maybe we have an abnormal escape of the neurotransmitter from the vesicles. So we have abnormal escape from the vesicles, from the secretory vesicle and the presynaptic terminal, or we have expulsion or evacuation of the neurotransmitter. The stored neurotransmitters, they are going to be evacuated and released, or we have inhibition of the enzymes or blockage of the enzyme or degradation of the enzymes that allow the degradation of the neurotransmitters. So these enzymes, they are blocked. No more function for these enzymes that are responsible for the degradation of this neurotransmitter, and this neurotransmitter will have a higher and a higher effect since it's not degraded by the enzyme, or the blockage of the recapture of this neurotransmitter. So these neurotransmitter, they are not recaptured. So they are very similar to the one that we started with that, unless for these two. So these two, they are a little bit different. So we have abnormal escape from the vesicles, where we have abnormal exocytosis from the neurotransmitters and the presynaptic terminal, or we have evacuation of the stored neurotransmitter, meaning that the stored neurotransmitter, they are going to be evacuated under abnormal conditions, or we have to inhibit or to block the function of the enzymes that degrade the neurotransmitters, or we're going to have to block or to prevent the recapture process, so that neurotransmitters, they are not recaptured by their enzymes or by the presynaptic terminal, and they are not degraded by the enzymes. So these disorders, they have amplifying effect. What does this mean? They have higher effect of the neurotransmitter. So neurotransmitter هيضلوا يشتغل normally، هيصير يشتغلوا أكثر من قبل، more than the normal. Why? Because we have such disorders. So these disorders, they help in the amplification of the effect of this neurotransmitter. Then we're going to move to the disorders that lead to an inhibiting effect. So it inhibits the effect of the neurotransmitter. What are these disorders? First, inhibition of the enzymes that allows the synthesis of the neurotransmitter, or that leads or catalyze the synthesis. So these enzymes that catalyze or that synthesize the neurotransmitter, these are inhibited. So there is no more synthesis of the neurotransmitter. So we have inhibitory effect at this level. Or B, we have blockage of the liberation of the neurotransmitter from the postsynaptic receptors. So they are blocked, or blockage of the receptor sites. So they reach the postsynaptic membrane, but the receptors, they are blocked. They are occupied by other substances. Like for example, let's say the acetylcholine and the GABA. Whenever the GABA binds, so or the curare, for example, bind, so they have an opposite effect. So whenever they bind, they have highly similar spatial configuration. So they bind, so the blockage of the receptor sites will occur. And this is an inhibitory effect. So this is related to the disorders either with amplifying effect that lead to the increasing of the effect of the neurotransmitter, or the disorders that lead to the inhibitory effect that inhibits the function of the neurotransmitter. So for a neurotransmitter to have an amplifying effect, what should it be? It should be a higher effect of the neurotransmitter than the normal. So maybe this neurotransmitter is not degraded by the enzyme. Maybe this neurotransmitter is not recaptured. Maybe this neurotransmitter, they escape from the secretory vesicle in the presynaptic terminal under abnormal condition. While inhibitory effect, inhibiting the action of this neurotransmitter. How? Maybe blockage of the receptor sites. Maybe the synthesis of the neurotransmitter is not enough. How? Why? Because the enzyme that helps in the synthesis process, it's blocked or inhibited. Or blockage of the neurotransmitter liberation. So all of them, they have a role in inhibiting the function of the neurotransmitter. So as a conclusion, in this document, what do you have to know? You have to know what is the agonist antagonist. You have to know that the acetylcholine and curare, they have opposite function. They are antagonists. We said that the curare, it causes the relaxation of the muscle, while acetylcholine, it causes the contraction of the muscle. Also, we talk about the enkephalin and the morphine. Both of them, they inhibit the pain circuit by inhibiting the exocytosis of the P substance, either partially or totally. So they are agonists. Enkephalin, they are endogenous, while morphine is an exogenous. Endogenous is produced naturally inside the body, while exogenous it's provided from an external source outside the body. This is relating to the enkephalin and the morphine. You have to know them. So these are agonists. They inhibit the exocytosis of the P substance either totally or partially. Morphine has total inhibition of the exocytosis, and we talk about this in the pain circuit in the introduction for the nervous system. We talk about morphine and about enkephalin. Also, you have to know about this table. Curare, it works about the fixation of the neurotransmitter on the postsynaptic membrane. Cocaine, it blocks the recapture or it inhibits the recapture of the dopamine by the presynaptic terminal or presynaptic membrane. Amphetamine, they facilitate the release of dopamine from the presynaptic neuron. And the benzodiazepines, they have a role in facilitating the fixation of the neurotransmitter GABA. Also, we talk about this part 4 disorders that lead either to amplifying effect or to inhibitory effect. So this is everything for document 4. Here, chapter 12 is almost done. So we have these two documents, document 4 and document 1, neurotransmitter and membrane channels. And this is everything for chapter 12 for this session.